The overall goal of this procedure is to generate high-quality cultures of cardiomyocytes from pluripotent stem cells and to appropriately characterize the end product using flow cytometry. To accomplish this, high-quality monolayer cultures of undifferentiated pluripotent stem cells are generated. Cardiomyogenesis is then induced by modulating the Wnt pathway using small molecules. Next, the cardiomyocytes are fixed, permeabilized, and stained using conditions that are specifically optimized for each antibody. Finally, the cells are analyzed by flow cytometry. Ultimately, results can be obtained that show cultures that are highly positive for cardiac markers TNNI3 and IRX4 and demonstrate ventricular-like action potentials.
The main advantages of this technique over existing methods, like those that employ unbraided bodies, are that it is relatively simple to implement and is cost-effective. Moreover, the technique generates cultures of cells that are highly positive for cardiac troponin I without the interline variation in efficiency observed by other protocols. Visual demonstration of the method is critical, as the pre-differentiation cell quality will significantly affect differentiation efficiency. Also, improper fixation and permeabilization prior to antibody staining will lead to suboptimal flow cytometry results. Demonstrating the procedure will be Subarna Bhattacharya, a graduate student from my laboratory.
The most challenging aspect of the following procedure is maintaining high-quality, undifferentiated pluripotent stem cells prior to differentiation. The use of cells with suboptimal growth characteristics will